All right, so I have figured out a couple of new tricks. I got my face in here, so now you have to look at that whenever I'm making these videos. And also, I've got two screens over here, so I'm kind of looking over to the side periodically as I'm looking at my terminal. So in this video, I am largely demonstrating assembly. So we have our reads already made. So we're going to assembly these reads, right? So we have during the sequencing step, what happens is that these transcripts they get cut up into small fragments, say 150, 300 bases, whatever. And when we sequence them, we don't know where they go, and we don't necessarily know which gene they came from. But what we can do is look for overlapping reads with sequence or identical sequences that overlap. Um, it gets a lot fancier than that, and I'm going to post a couple of links about assemblers and De Bruin graphs in the description. Um, I encourage you all to look at it, although it's not entirely necessary to understand the nitty gritty details behind it. Um, it is a good idea to at least, I guess, see these words and see kind of what's happening. But yeah, it's probably a little bit more important of knowing how to assemble and the best tools to do so rather than we're not developers, we don't necessarily need to know what's going on behind the scenes. Um, so let's look at our test data that we've been messing with, that I've been messing around with y'all as an example. um, and when we left off, right, so um, we ran Trimmomatic to have a clean set of reads, right, to reduce the garbage out of our reads, because if we have garbage, then our assembly will be a weaker quality, it won't be as good. So we need to get rid of crap reads. Um, at this point, we can get rid of these the unpaired stuff because I'm not going to use them in assemblies. I usually don't use them. I'm just going to stick with the clean reads. Let's see. The software we're using is in Trinity. And so this is the main software that's used for Symbol. This is probably the go-to RNA-seq assembly software. It's called Trinity. Um, there's a lot of stuff going on in the behind the scenes. Um, but yeah, the main tool, the main wrapper, what's controlling everything is this Perl script called Trinity. But it's going to interact with a bunch of tools in these directories. And so we kind of have to call Trinity from its source, right? We can't just call Trinity and it'll work. We have to kind of call Trinity from its source. At least it's because Trinity is in options. Um, it's probably a little bit easier to just call it from its home. So let's go back to the reads. Okay. Okay. So whenever we call Trinity, we're calling it from op Trinity, and then Trinity is the main Perl wrapper. And so we're going to get a fairly intuitive, a fairly helpful help menu when we just run Trinity by itself. So we have to have these four major parameters in here. We have to tell Trinity what the sequence type is, whether it's a FASTA or FASTQ. We have to allocate how much memory will be used. So assemblers are probably the most memory hungry tools in all of bioinformatics, right? If we're trying to assemble the entire human genome, we effectively need like 500 gigabytes of memory to do so. Thankfully, transcriptomes are considerably smaller, right? They're only 1% or 1 to 2% of an average genome or a vertebrate genome. So we don't need so much memory with transcriptome assemblies. And early in this semester, I wanted you all to do a full genome assembly on something small like E. coli, something along those lines. But we kind of ran out of time, and so I'm kind of killing two birds with one stone, both the concept of assembly and using assembly techniques to reconstruct all of the genes that are expressed from a tissue. So um, yeah, so we have to again we have to have the sequence types, the max amount of memory, the left. So we have to decide. We have to tell Trinity which is our left-hand file and which is our right-hand file, or which is our forward-facing reads, which is our reverse-facing reads. And then we also have a couple of miscellaneous options that are fairly helpful, right? So CPUs, we want to assign a fair amount of CPUs because uh, this can be also very or compute heavy. There's going to be a lot of processes that can happen in parallel, so we want we'll probably set it to 12. Um, let's see, we'll set this SS library type. So how RNA-seq libraries are generated is like uh, not enough time to explain, but um, we can ultimately, if we set this parameter, if we set this option and it's right, we can generally get a better assembly. At least all the genes will be in the 5 prime to 3 prime orientation, right? That's the whole point of this, is trying to get all of our genes in the 5 prime to 3 prime because they're expressed 5 prime to 3 prime. But that doesn't necessarily mean whenever we assemble everything that these genes will be in the 5 to 3 prime, and setting this SSLib type should help that out a little bit, and hopefully I'm not making that up. A nice little thing about Trinity, again, this is kind of the go-to software, is that it does have this trim-o-matic option. So you can set a trim-o-matic line. So if you have our raw reads, um, we can set some clipping parameters, like the sliding window and the Illumina adapter clip and etc. And so it will Trinity will run Trim-O-Matic first and then assemble from the clean output reads, right? So that is kind of nice. This output by default, what Trinity will do is make this full output directory, and it's going to have literally every single document that it makes stored inside of it. I think the default is like Trinity out. I can test that. Yeah, the default is Trinity out dir. But we can assign it to anything we want. Although it does recommend that um, you keep Trinity somewhere in this name. Otherwise, the weird things may happen. Another nice option, we're not going to run it this time, is this full cleanup, right? So this I already did this once on my clean reads that I've already made, so I can see how much space this directory is taking up by using du-h. Ultimately, so this whole directory, just storing all this information, costs 22 gigabytes of space, and most of it's garbage that we'll never ever look at. So, um, right, so to prevent having all of that extra garbage, we can set this full cleanup option, and so that's all that's going to remain is just the assembled transcripts when all is said and done. All right. Um, again, this is very a nice helpful help menu to help you figure out what files need to go where. Right, here's an example line of everything that you need for a paired in reads, seek type, max memory, left, right, and the amount of CPUs. That's literally all you really need to do to actually run this piece of software. All right. So since this is going to take a while, I'm going to run this in a teamup shell. Um, right, this one will take hours. It doesn't even matter if it's a small file. This will still take several hours for Trinity to finish. So let's see. So opt Trinity. Um, I'm not going to make a script of this because there aren't so many lines with it, and we just put it in. We may make a script of this eventually, um, but for now, just to run the line, we'll do it this way. Seek type is FQ. Max. I'm gonna have to hold on. Hold on. I need my help menu to know what my options are. All right, so I need seek type, FQ, max, memory. I'm going to set this to 50 gigabytes. That's probably fair. I mean, it could probably be 20 or so, but just for safety, I'll leave it at 50. Our left is going to be the clean underscore 1. The right will be the clean underscore 2. We'll set CPU to 6 and SS. Oh boy. Hold on, I'm going to detach for a second. SS libtype. SS libtype. And so this is going to be RF. And we set it as RF. If we have a live conversation about this, I'll talk about it then, but I kind of don't want to talk about it now. CPUs, I'm going to set this to 12. And hopefully click go and everything's going to run well. So this did take me, I wanted to make a video yesterday, but this did take me a couple of hours to actually set up Trinity and all of its dependencies to make this work as well as it can. So this is going to run, and again, this will take a while, but what it's going to do is that it's going to break up these reads. It's going to, convert them to fast Q to fast A. I can detach from this and kind of look through some output that I already made earlier. So I already ran this program on this software, on these reads, and I have this output, right? So I have this whole amount of output here. And so we have like this, let's see, we have a write file. Nope, we don't, not anymore. We have this both.fa, and what this both.fa is that it keeps all of the reads. It's going to convert all of these FASTQ reads into fast A, right? And also, what's after that happens, it's going to run this program called Jellyfish. And Jellyfish, what it's going to do is it's going to scan through all of these reads, and it's going to look for and count all of the unique reads that are 25 bases long. And so we don't need to necessarily look at all of Jellyfish, but I can less it as it's being counted out. Right, so all of these reads, so what this is saying is that this sequence of A's 25 base pairs long was found about 17,000 times. Right, that's bound to happen. It's an RNA-seq library. These things have poly-A tails, so you're going to see a lot of poly-A's. No surprise there. But right, these are all of the unique sequences that are 25 base pairs long. And Trinity will do some magic with these sequences, but it has more to do with the de Bruijn graph construction. Again, I'll have a video about that because I honestly, I'm not good enough to explain it. I can use this tool, but besides the concept of what it's doing, I have no idea what it's actually doing behind the scenes. But either way, it's counting all these reads, right? Anytime we see a -2, it just means that this read was present, or that sequence, that mer. It's also called a mer was present 2 times. This one was present 4 times, etc. All right, when all is said and done, the only file that really matters out of this output is the trinity.fasta. Right, so this actually is our assembled transcripts. Right, so it's going to keep all of these in place. And you'll see not all of them start with a start codon. Most of them won't start with a start codon because it's doing the whole transcript, right? And so there's a 5' UTR, there's a 3' UTR, and between those we'll have the actual CDS. And within that CDS, we'll see start and stop codons. But we won't always see everything because it's just an artifact of transcriptome assemblies. Not every transcript will be 100% complete. And again, not every transcript will be 100% identical. For example, so if I rep-c on the open char, so I'm counting the number of sequences in this file. Let's see, there are 125,000 sequences. But you could predict there's within the liver tissue, there's probably only 10,000 genes expressed and maybe 5 or so unique transcripts per gene. And so these transcriptome assemblies, they tend to have some artifacts, and we usually only want to filter for things that we can recognize, particularly like CDS sequences and things like that. But I think I'm going to call it there for now. This is just running. This is just performing the transcriptome assembly. Take a look, see if you can get this done and working. I think running Trinity is a little bit easier than running Trimmatic. It's just an easier help menu. Yeah, I think that's it. So yeah, this is going to take hours. I'm just going to kill it now because I've already done it. But when all is said and done, you should have a Trinity output directory that has everything you want in it. And next time we run this, we'll add some parameters so we don't have this massive 22 gigabyte directory output. We'll just have the fastest sequences to save space because we kind of are quickly losing space. And then whenever everybody runs Trinity on all this stuff, we'll definitely be out of space pretty quick. All right. Good luck. And until next time.